So she will be joining. She's saying some trying to connect.
Yes.
Yeah. Dr. Kunal, you can start.
Yeah. My screen is visible, Ashish ji, full screen.
Yes.
Yes, yes. Visible, visible.
Good evening, all delegates on this global webinar, PACLIOL, sponsored by Panacea Biotech Oncology Division. Today's topic is the successful R&D journey of nanoalbumin-bound paclitaxel and an immense role in the management of metastatic breast cancer, CA pancreas, and NSCLC. Panacea Biotech is the first company who has attempted and successfully manufactured and marketed nanoalbumin-bound paclitaxel, which was a great challenge across the world. This challenge to overcome, it took a lot of efforts, number of R&D scientists working together for many number of years, and many experimental studies, molecular biological innovation intervention, sourcing the materials and methods across the globe, and manufacturing it successfully, doing the clinical research and regulatory required clinical trials, experimental studies, and human studies, which has made the first approval by the regulators to Panacea Biotech and awarded in 2011 the marketing approval, which has got warm welcome by oncologists across the country. Each and every oncologist from India has welcomed because it has reduced the cost of the metastatic breast cancer more than half. Affordability and quality medicine, which is the principle of Panacea Biotech, has got fulfilled. With this, I realize that the Panacea Biotech R&D and the medical and marketing team, which has got accolades, acceptance, and complete confidence by oncologists to use Nab-paclitaxel. To know this successful journey, we have today with us the R&D scientist, Dr. Naveen Jain, who is alumni of the Punjab University, who studied in a USA for the molecular technology and molecular biology, and who is the one of the important lead of the team which has successfully manufactured and marketed Nab-paclitaxel. And it is very interesting to know from him what is the journey of this and why the doctors are confident to replace the originator Abraxane and to use the PACLIOL. Today for the session, we have Dr. T. Raja, Dr. Niti Raizada, and Dr. Govind Babu. For Dr. T. Raja, everyone knows across the country, sir is the teacher, guide for every oncologist in the country and the doctors, those who have studied here and went for the further education abroad. He's a director of medical oncology currently at Apollo Hospital, head of department in medical oncology, Apollo Proton Cancer Center at Chennai, and he is leading the various roles and important decisions for the hospital, for the government, and for the Oncology Society of India. He is the pillar, and we will get enriched about to understand that the Nab-paclitaxel, its clinical trials, its research, and its important place in managing cancer, CA pancreas. Dr. Niti Raizada, the Director, Medical Oncology and Hemato-Oncology of Fortis Group of Hospitals from Bangalore. Ma'am is continuously into the management of the various oncology cases. She is the doctor continually believing on the Indian drugs, Indian generic Nab-paclitaxel, and consistent support. Ma'am is having the expertise into the managing the CA breast. The metastatic breast cancer is the area of topic of this immense interest. And we will see that how to place the Nab-paclitaxel along with other chemotherapeutic agents and interesting cases Dr. Niti Raizada ma'am will discuss with us. We have Dr. Govind Babu, the consultant medical oncologist at St. John's Medical College and Hospitals, Bangalore. He was the formerly professor and head of Division of Medical Oncology, Kidwai Memorial Institute at Bangalore, and President Indian Society of Medical and Pediatric Oncology. So his contribution as a global level is the President SARC Federation of Oncology and co-chair ISMO India Summit 2020. Dr. will enlighten us on the Nab-paclitaxel and SCLC evaluations with the clinical incidences, case presentation, and interestingly, Sir will tell us about his own experience of SCLC. I request to begin the session to enlighten us the interesting journey. This is almost the 1 decade after the successful launch of Nab-paclitaxel. Dr. Naveen Jain, I'm requesting you to enlighten us on the Nab-paclitaxel Pacliol of Panacea Biotech. Over to Dr. Naveen Jain.
Yeah, thank you, Dr. Kunal.
Sir, you can share your screen.
Yeah, is it visible?
Yes, sir, very well.
Yeah, thank you, Dr. Kunal, and good evening to all for this global webinar on the Nab-paclitaxel. So I will take you through our drug discovery and drug development journey for developing first complex generics of Nab-paclitaxel, that is Pacliol. So my presentation will include the brief summarization of the importance of the nanotechnology, why nanotechnology is quite interesting for the application for developing the Pacliol, and its comparative advantages over the conventional paclitaxel, and our journey to demonstrate the pharmaceutical and bioequivalence similarity with Pacliol and Abraxane. In that journey, I will also highlight our CMC development part, preclinical development part, including GLP toxicity studies and bioequivalence studies in the metastatic breast cancer patients in comparison to the innovator, that is the Abraxane. So as we know, nanotechnology has the immense application in the developing the targeted drug delivery system. So in the Panacea Biotech, what we did, we identified the problem in the conventional paclitaxel, that is the solubilization. As we know, conventional paclitaxel uses the chemophore to solubilize, and this chemophore has the several side effect profile, anaphylactic reactions, and a lot of pre-medication is required due to this chemophore. And the infusion time is also prolonged due to the chemophore because we have to infuse very, very slowly. So by this virtue of the nanotechnology, we reduced the particle size and clubbed this paclitaxel with the human serum albumin and avoided the need of the chemophore. But the challenge is that development of the nanosize of paclitaxel in the R&D setup and successfully transferred to the production scale. It needs the lot of synchronized efforts with the isolator requirement, with the aseptic manufacturing facility, with the negligible risk for the exposure to the operators and users. These are the basic steps. I'm just highlighting how the nanoparticles is getting manufactured and leading to the lyophilization at the production level. So we developed it. You can see the pictures of the manufacturing setup facility where you can see that this aseptic atmosphere as well as the isolators where people can work independently without exposing to themselves to the paclitaxel, which is chemotherapy. And the manufacturing facility also has the robust and validated in-process quality control systems and the stability chambers, as well as the characterization facility for the nanoparticles. If we compare the conventional and Nab-paclitaxel, as we know, the advantage of the paclitaxel is that we can infuse higher amount of dose, that is a 260 milligram per meter square, and there is a very short infusion time, that is only 30 minutes, and no need of the that is a chemophore type of solvents, because it has contained the human serum albumin and easily can be reconstituted with the saline, and there is no need of the pre-medications like corticosteroid and antihistamine. And thus the biggest advantage as well is that tumor targeting, because the majority of the tumor spark protein is expressed. So this albumin has the higher affinity for the spark protein, so it can be delivered directly to the spark protein, which is overexpressed in majority of the tumor. So while in the development, these are the parameters we have first optimized. What are the parameters which needs to optimized? Appearance, identifications, assay, albumin content, water content, reconstitution time, impurity profile, osmolality, pH, residual solvent, endotoxin levels, and particulate matter. These parameters are at macro level. Beside the characterization of the nanoparticles, the characterization of the nanoparticles, which are the particle size distribution, particle morphology, surface potential, crystallinity of the paclitaxel, fraction of the bound and unbound paclitaxel after the reconstitution, and fraction of the albumin bound and unbound after the reconstitution, and in vitro release kinetics. So these parameters are the dedicatedly developed and validated for the characterization of nanoparticles in our facility and demonstrated successfully in comparison to the XA. This CMC development, after the development of the proof of concept in the CMC, then we developed the lead formulations, which has the significant equivalent profile in comparison to Abraxane. Then we started working on the preclinical development. Preclinical development includes the demonstration of the efficacy of the Paclitaxel in the tumor xenograft model, and the tissue distribution, and the plasma, and the tumor release profile. So we took MX1 tumor xenograft model and injected into the mouse, skid mouse, and observed the tumor regression profile up to the 36 days. You can see in the graph the tumor regression profile is comparative in equivalence with the marketed product, that is a Abraxane. Then we took also ST29, that is a colon cancer cell model. We implanted into the skid mice this ST29 tumor xenograft and observed the comparative tumor regression profile into up to 8 days, and you can see the regression profile is exactly equivalent to the innovator Abraxane. Then we also looked into its distribution into the plasma and blood, and in the biopsy mice, we injected this formulation, Paclitaxel and Abraxane, and bioequivalence was observed in this mouse study. Then we also observed the equivalence in the tissue distribution in the biopsy mice. Why it is so important? Because nanoparticles behaves very differently in each organ, and because the particle size matters. If the particle size is larger than the respective organ uptake, then it will be uptaken by the liver. Majority of it will be uptaken by the RES system. So we took the 5 major vital organs and observed the tissue distribution in each organ post injection of the 5 minutes and 6 hours, and we observed the exact equivalent tissue distribution profile in the biopsy mice. Then the next step was that how it is behaving in the tumor-bearing mice in terms of the pharmacokinetics and the tumor distribution. So we observed, we implanted MCF7. Again, it is a breast cancer tumor xenograft model, and we injected Paclitaxel and Abraxane into the mouse and observed the plasma plasma concentration as 6 hours and tumor concentration at 24 hours. We measure that plasma and tumor retention is exactly equivalent into this MCF7 tumor implanted xenograft model. Then, after successful demonstration of the efficacy and pharmacokinetics and tissue distribution as a part of the drug discovery and drug development, we need to demonstrate as well as tolerability and toxicity profile at high doses into the animal species as per the drugs and cosmetics. So we took two type of the studies, single dose, repeat dose toxicology studies, and in the comparative arm, we put the Abraxane, and we found our toxicity profile and safety profile is exactly comparable to the innovator, that is Abraxane, in the GLP setting. So it was a successful demonstration in the preclinical system as well as the pharmaceutical similarity. Now we took further ahead and demonstrated the bioequivalence into the clinical setting. In this clinical trial, what we did was a bioequivalence study. It was open label, 2-period, 2-treatment, 2-sequence, single dose crossover bioequivalence study with the paclitaxel and in comparison the which is manufactured by the Celgene Incorporation. So we took around 90 patients, enrolled 90 patients, that is a metastatic breast cancer patient, and the study was completed into the 78 patient and injected the doses 260 milligram per meter square as per the clinical protocol up to the 30 minutes of the infusion time. And after the post injection, around 22 samples was collected to demonstrate the bioequivalence. This study was conducted at various centers across the India, and to demonstrate the bioequivalence, we took two parameters in the blood: unbound paclitaxel and total paclitaxel. Unbound paclitaxel, we took there is a standard parameters of the bioequivalence is defined by the FDA, that is a Cmax under the curve at the T or infinitive. So in all these three parameters, our product was bioequivalent within the 80 to 120% of the criteria of bioequivalence with the unbound paclitaxel. You can see this graph that product was exactly bioequivalent in the unbound paclitaxel. And then we also monitored the total paclitaxel at the 90% of the confidence interval, and all three parameters, Cmax, area under the curve, T and infinitive, the product was exactly bio. If you look into the T by R ratio, it is exactly similar with the within the 80 to 100%, 125% of the bioequivalence criteria. So after the successful characterization, CMC, pharmaceutical equivalence demonstration, preclinical demonstration, and the bioequivalence demonstration in the metastatic breast cancer patient, this product was successfully launched in the India, and this is the first characterized Indian Nab-paclitaxel product, which has all the advantage over the conventional paclitaxel type, like there is no need of pre-medication, lesser infusion time, and it is bioequivalent to the innovator. I'm very pleased to share that this product has been already registered in the Australia and Canada by Health Canada, and we are awaiting the approval of this product from the US FDA very soon. And after launching this product in India, we received the next year only the Biospectrum Product of the Year award for the making the indigenous technology an affordable product to the Indian metastatic breast cancer patient. This is the team in our R&D scientist team, which has significantly contributed in the development of this product, from the material sourcing to the preclinical development, CMC characterization, and the clinical characterization. And this is the facility, which is the state-of-the-art facility, which has all the requirement of the nanoparticle characterization, validations, optimization, and successful technology transfer to our art manufacturing, US FDA approved manufacturing facility at Baddi, Himachal. Thank you to you all for hearing this drug discovery and drug development experience of the developing the Nab-paclitaxel.
Thank you very much, Dr. Naveen Jain, for nicely elaborating the journey of inventing the first generic, global generic of Nab-paclitaxel. Its various preclinical studies and nicely showing the R&D center where you have developed and manufactured it. Many more questions will come definitely that why the supply of Pacliol will be disinterrupted later on, what was the challenges company was facing, and why the other pharmaceutical companies were not able to manufacture the right quality or equivalent to the originator or Nab-paclitaxel of Panacea Biotic. And still many companies are struggling. What are the challenges and difficulties will be get discussed in a panel discussion. I invite Dr. T. Raja to enlighten us on the first systemic therapy in CA pancreas, first line, second line therapy, and BRCA mutations, and also the important points what Dr. Sir wants to elaborate to the audience. Over to Dr. T. Raja.
Thank you so much, Panacea, and thank you so much, all of you, Dr. Naveen. Excellent lecture, because we could understand how much difficult it is to develop something and a quality product, and so much of hard work goes behind it. We truly appreciate it. Allow me to share my slide. So I hope that the slides are visible and I'm audible.
Yes, sir. It is very well visible and you are very clearly audible. Please continue.
Thank you. So, thanks for asking me to speak on CA pancreas, and particularly the topic given to me is first line, second line, and BRCA mutations, and whatever, anything more on CA pancreas. That is my assigned topic today. And let me begin with my heartfelt tribute to this gentleman. I think most of you may know him, but some of you may not know him. He's a legendary playback singer, and we lost him yesterday here in Chennai. And he has been an epitome of something what we just now heard. He is an epitome of talent, but then after that, immense hard work. Over nearly 40 to 45 years, he dominated the field, and he has been an iconic figure in producing some of the best numbers of all time. And Mr. Ilayaraja would go on to say only one word, that please come back. And Mr. Rahman would say that I am devastated. So they simply express the nation's feelings towards him. So we'll go to the topic: metastatic pancreatic cancer, first line treatment. What is the clinical evidence of what we have? Erstwhile, it was always gemcitabine in pancreatic cancer, be it metastatic or be it early. So anything that has to be studied has to be pitched against the standard of what we call gemcitabine standard. FOLFIRINOX is an important combination, and in a phase 3 trial, multi-center randomized phase 3 trial, FOLFIRINOX versus gemcitabine for patients with metastatic pancreatic cancer was studied. And what do we learn out of that? This paper came about 7-8 years ago, and it was shown that whereas the PFS for single agent gemcitabine will be 3.3 months in the metastatic setting, FOLFIRINOX would achieve a PFS of 6.4 months, and a median overall survival would improve from a 6.8 months single agent gemcitabine to 11.1 months with FOLFIRINOX. A good PFS and OS advantage with the FOLFIRINOX combination. Fortunately, the safety profile was not very difficult with FOLFIRINOX, even though there were higher incidences of neutropenia, febrile neutropenia, and thrombocytopenia with FOLFIRINOX, but still largely manageable safety profile. And the quality of life, paradoxically, was either same or better than single agent gemcitabine, despite the toxicity profile that we know. Many things like global health status and cognitive and fatigue, nausea, and were all found to be much better with FOLFIRINOX. And therefore, we understood that FOLFIRINOX is a combination that can probably be superior to single agent gemcitabine in the metastatic breast cancer. But then gemcitabine's work was also further elaborated by, doublets. Gemcitabine combinations were also explored, and there were several phase 3 trials once again on combinations of gemcitabine-based therapies. Particularly to note is gemcitabine oxaliplatin, which would give a median overall survival of 9 months as compared to 6 to 7 months with single agent gemcitabine. Gemcitabine capecitabine, 7.1 months median overall survival, and very important to note that gemcitabine plus nab-paclitaxel also could reach the 8.7 to 9 months figure as compared to 6 to 7 months of single agent gemcitabine. So these were the combos that improved the performance of single agent gemcitabine. An important trial, impact trial, wherein gemcitabine plus nab-paclitaxel doublet, again multi-center phase 3 trial against gemcitabine single agent, and the primary endpoints were OS and PFS. What was seen with the gemcitabine plus nab-paclitaxel combination was a superior PFS of 5.5 months and a superior OS of 8.5 to 8.7 months with very good hazard ratios. And again, very acceptable safety profile with the doublet as compared to single agent gemcitabine. So these two studies, the FOLFIRINOX phase 3 trial and the IMPACT phase 3 trial, showed that there can be two important frontline options for patients with metastatic pancreatic cancer, which can be better than single agent gemcitabine. If you put them together and see head to head, the trials were equally strength of equal strength, and they were comparable from the numerical point of view. If you look at the ORR and the disease control rate, PFS and OS, you would see that numerically the FOLFIRINOX trial showed a slightly superior numbers as compared to the nab-paclitaxel gemcitabine IMPACT trial, but this is not exactly a head-to-head comparison between the two possible combos. Both were independently compared against a single-agent gemcitabine. So for first-line chemotherapy regimens, now at least we have a couple of choices. So how do we make a decision? We need to look at the performance status of the patient. We need to look at the age of the patient, and you would definitely consider the comorbidities. For example, if there is a pre-existing cardiac condition or if there is a significant sensory neuropathy, you probably would lean towards a gemcitabine nab-paclitaxel combination. Or in a well-fit individual who can tolerate infusional regimen, you probably would lean towards a FOLFIRINOX. Coming to second line treatment options in metastatic pancreatic cancer. This picture also shows, Mr. Rajinikanth paying tribute to Mr. Balasubramanian, and you would know that Rajinikanth has delivered innumerable hits. And what is also less known is that most of his intro songs were always sung by Balasubramanian. Rajinikanth was so particular that any of his film intros should be done by Mr. SPB, and wherever he has delivered the intro song, that film has become a blockbuster. So that is his sentiment. Coming to the second line options in metastatic pancreatic cancer, we now understand that at least half of the patient, 50% of the patient would eventually be a candidate for second line treatment post progression. Until recently, there is not much options when patients progress on first line, and people would try something like a oxaliplatin combos. That were the way to go. And there are at least two good phase 3 experience again with second line chemotherapy with oxaliplatin based combos. One is Pancreox, in which modified FOLFIRINOX was compared against a single agent 5-fluorovarin, and the other one was Conco 003, in which oxaliplatin 5-fluorouracil infusional regimen was compared again with 5-fluorovarin. And in both these instances, the first line therapy was gemcitabine. So in patients who have progressed gemcitabine, you now have two options. Either you can use modified FOLFIRINOX or an infusional oxaliplatin based 5-FU regimen. The other option is irinotecan based therapy, nano liposomal irinotecan plus 5-FU leucovorin infusional regimen as compared to single agent 5-FU leucovorin. And this is again a phase 3 trial on patients who progress on gemcitabine. These are the OS outcome with the NAL-IRI protocol. This is the Napoli trial. NAL-IRI protocol was studied in a 3-armed way in which 5-FU leucovorin was the comparator arm, and the NAL-IRI and then NAL-IRI plus 5-FU. These were the two experimental arms, and NAL-IRI plus 5-FU leucovorin was definitely superior to single agent 5-FU leucovorin. So currently you have treatment sequencing options. If patients were already treated with prior therapies, you have some second line options. So how do we sequence? In the first line, gemcitabine based combination like gemcitabine nab-paclitaxel is a very good option, and in patients with a poor performance status, single agent gemcitabine is an option. In patients with a very good performance status, FOLFIRINOX is a good option, and in patients who progress, you have the nanoliposomal irinotecan based therapy, or you have, in patients who have progressed on FOLFIRINOX, you have the gem nab-paclitaxel combination as an option. Coming to the molecular profiling of pancreatic cancer, what do we see is that there are several molecular changes that can be picked up in the metastatic pancreatic setting. Either those molecular changes can be immunogenic in their pathophysiology, particularly the MSI and MMR defects, or it could be the BRCA1 and 2, PAL-B2. Basically, they are all DNA damage response, defective DNA damage response modifiers. And there can be some other rare abnormalities like NTRK fusions. And the BRCA mutation particularly is very important because it is seen in about 7% of patients with pancreatic cancer. And if you add all the additional mutations that can implicate the repair pathway like PAL-B2 and ATM, you can get around 10% of patients in pancreatic cancer who have some deficiency in that pathway. So in them, maintenance olaparib was looked at after the first line therapy with platinum-based therapy in metastatic pancreatic cancer. Olaparib was studied as a maintenance option as compared to placebo. Again, a very robust phase 3 trial in which nearly 154 patients were studied. And in those patients, it was shown that in patients who were put on maintenance olaparib post their first line therapy, after they achieve a good response or a very good partial response, olaparib can lead to a superior progression-free survival in the maintenance setting of 7.4 months as compared to 3.8 months in the placebo arm. There are now several ongoing studies with PARP inhibitors, which can be now combined with either irinotecan based therapy or oxaliplatin based therapy or nanoliposomal based therapy. All these studies are ongoing with PARP inhibitors. The Memorial Sloan Kettering experience would show that the genetic alterations in pancreatic cancers can be now looked at whether they have actionable genetic alterations or not, in which there were several actionable genetic alterations that had potential treatment options with a level 2B evidence to support, for example, the BRCA1 and 2, BRAF, V600E, and so on and so forth. And a level 3B evidence for therapeutic options are available for a PIK3CA and an FGFR1 and 2 amplification. And again, a level 4 evidence where there is some preclinical evidence, but not yet FDA approved biomarker or drug is seen in quite a bit of patients. If you look at level 2B, you have 5% options. And whereas in level 4, you have nearly 60% of patients expressing some sort of biomarkers where potentially in future, probably you will find a targetable drug. And this is the pembrolizumab work in patients who have a higher MSI, and that's the waterfall plots. And again, for the rarer NTRK fusion, larotrectinib can also show an impressive waterfall plots in solid tumors, including pancreatic cancer. And two of my panelists today are from Bangalore. And this is again to acknowledge the versatility of Mr. SPB's work. he has sung not only in the Tamil language, but he has been a notable performer in the Kannada language, and he has delivered innumerable hits in many South Indian languages. Coming to resectable pancreatic cancers, we look at them as resectable or borderline resectable or unresectable. Particularly in resectable pancreatic cancer, now we look at whether we can introduce a neoadjuvant therapy, and there are debates about pros and cons of neoadjuvant therapy, but we need to look at some of the important trials. The SWOG 1505 study, in which modified FOLFIRINOX and Gem-Nab-paclitaxel combos were both offered as neoadjuvant option, and after treatment, restaging, surgery, and then continued with these therapeutic options. And the summary of the results of this SWOG 1505, which is again a phase 3 randomized trial, you would see that at second year overall survival as a primary endpoint, the modified FOLFIRINOX would give us an OS of about 43%, and the Gem-Nab-paclitaxel would give an OS of about 46%. Independently, both of these look good, but unfortunately, the primary endpoint was set at 2-year OS of about 58%. Therefore, statistically, this study was deemed to be not a positive study, but there is a lot of take home from this study, because the investigators showed that there was a very rapid accrual in this trial, indicating that there is a growing appetite for preoperative treatment options with pancreatic cancers. Japanese have done a lot of work on neoadjuvant chemotherapy. This is the gemcitabine plus S1 work on neoadjuvant chemotherapy, followed by surgery, neoadjuvant chemoradiotherapy, gemcitabine radiation, and then gemcitabine and surgery. So neoadjuvant chemoradiation is another option. This is a PREOPANC study. All are robust phase 3 studies supporting the concept of neoadjuvant option and in resectable pancreatic cancer. What about adjuvant strategies? There are again several important work in the adjuvant setting. There are randomized postoperative adjuvant work with single agent gemcitabine plus radiation and gemcitabine plus erlotinib, and the SPAC is gemcitabine plus capecitabine, again a phase 3 study for resected pancreatic cancer, and the PRODIGE is the adjuvant mFOLFIRINOX plus versus gemcitabine in the adjuvant setting, which is again a phase 3 trial. And the PRODIGE would show that mFOLFIRINOX is superior to single agent gemcitabine in the adjuvant setting also. And again, an important work, APACT, adjuvant Nab-paclitaxel plus gemcitabine in the adjuvant setting in resected pancreatic cancer. Again, a phase 3 trial in which nearly 800 patients were looked at in both arms, where the Nab-paclitaxel gemcitabine was compared against a single agent gemcitabine, again showing that you can achieve a numerical superiority with the DFS, and the median OS was found to be numerically superior, but the hazard ratios were to the tune of 0.82 and 0.88. These are important work, even though they have not exactly met the statistical endpoints, but these things show that one can improve upon the single agent gemcitabine with the gemcitabine Nab-paclitaxel in the resected adjuvant setting also. Fortunately, the Gem-Nab-paclitaxel again was not found to be too toxic. It was acceptable safety, with an acceptable safety profile in this setting. Again, a very useful therapeutic armamentarium in patients who have a good fitness scale in front of you. There are several randomized trials that are emerging. Again, one of the important trials that we would want to bring is perioperative modified FOLFIRINOX versus perioperative gemcitabine Nab-paclitaxel, and this trial is ongoing, and recruiting this would tell us whether we can use Gem-Nab-paclitaxel in the perioperative surgery and postoperative setting, and so on. So what are the conclusions as far as what is happening in the field of pancreatic cancer? In the early stage resectable disease, there is now phase 3 evidence to support that FOLFIRINOX is an important option for you. And again, neoadjuvant therapy is becoming increasingly popular. And in the metastatic setting, frontline option and second line options are now well defined. FOLFIRINOX is an important option. Nab-paclitaxel is an important option which have demonstrated survival benefit as compared to single-agent gemcitabine alone in phase 3 trial. And there are now important second-line options for you to consider. And with regard to the germline testing and with regard to your molecular profiling, you would consider germline testing in probably all patients with pancreatic cancer, newly diagnosed pancreatic cancer, regardless of family history, because you are going to see around 7 to 10% of patients in which there is a possibility of using maintenance PARP inhibitors. Again, somatic testing in the form of an NGS can be used for patients who have progression in which you can find some occasional mutation, genetic alterations, which are targetable either with immune checkpoint inhibitors or with emerging drugs like larotrectinib. So these are the advances in the field of pancreatic cancer. I've given you an overview, and hopefully I know that has given you something to think about and take home. Finally, ending again with the tribute to this gentleman. He has sung more than 40,000 songs in 16 languages, and a true genius. And he has done one thing: he has connected the country, all India together. Thanks to him, and thanks to Panesia for this opportunity. Thanks for all of you to listening to me.
Thank you very much, sir. Such a nice and elaborative session by you, which reflects that how you are pursuing life with science and art. You have started with the AMPEX study, which is the first approval for the US FDA for pancreatic cancer for nab-paclitaxel. Then you have taken through us the GEM-NAB and the gemcitabine and the FOLFIRINOX comparative study and outcome. You have also elaborated on the ongoing studies and the studies which are impacting resectable and non-resectable CA pancreas. And on every slide, you have shown that how the SPP has, touched, moved, and inspired the life of the artist on this screen. Similarly, in your own life, you are making us, and this is the first sessions where the R&D scientist is presenting the successful journey of Paclitaxel on this platform. And this has happened because of you. And we are happy with this succession of the session. I invite Dr. Niti Raizada to enlighten us on metastatic breast cancer, importantly, the place of nab-paclitaxel along with other chemotherapeutic agents and the interesting cases ma'am can describe to the audience. Over to Dr. Niti Raizada, ma'am.
Thank you very much. Good evening to everyone, and really appreciate Panesia's effort for putting all of us together to discuss on this Saturday evening. Thanks, Dr. Raja. It was an excellent presentation, and it was very concise and very informative for us to take CA pancreas forward. So what I'll be doing is I'll be talking on metastatic breast cancer management, but my focus will only be on nab-paclitaxel as a single agent or in combination, because we all know metastatic breast cancer is an infinite, unlimited discussion, and we will just focus on today's area of interest. So talking about just metastatic breast cancer in general, and especially the disease burden in our country. So globally, we know that amongst all cancer, breast cancer forms incidence of about 12%. And the mortality is almost half of this. So 6.6% of all the diagnosed breast cancer die in the subsequent year. And the invasive breast cancer incidence as in 2018 was about 2 million new cases. So as far as India is concerned, we have incidence which is much higher than globally. Almost 14% of all cancers in women is breast cancer. We have about 162,000 cases diagnosed annually, and more than 50% of these die of the same cancer, which is much higher in developing countries than in developed countries. In developed countries, only about 35 to 40% would die of breast cancer. And in our country, the peak of breast cancer also happens earlier than the Western population. So we have a peak which is in early 30s. Sorry, we begin in early 30s and peaks around late 40s or early 50s. 50 to 64 is the average age. And in the urban population, 1 in 22 women would be diagnosed as breast cancer. I personally feel there's an underreported statement. And in reality, across the globe, 1 in 8 women would develop breast cancer. And I think going forward, once the statistics is right, we will have almost similar figures in our country, what we actually see in our practice. So just going briefly about Nab-paclitaxel and overview, Dr. Naveen has discussed in detail, also talking about the bioequivalent study. But Nab-paclitaxel is an albumin-bound nanoparticle formulation, which is solvent-free colloidal suspension of paclitaxel and human serum albumin. Now, nanoparticle delivery system basically eliminates the need of toxic solvents like tremefor, which are normally done in the conventional paclitaxel by binding paclitaxel to albumin. This basically eliminates the need of pre-medication and can be given in as short a time as 30 minutes. Albumin binding through glycoprotein receptors, GP60, is the major factor. In metastatic breast cancer, there are indications of Nab-paclitaxel across all subtypes of metastatic breast cancer. We commonly use it in triple negative subset, but we also use it in hormone positive or HER2 positive subset. And we have also seen that it has clinical benefit once 2 or 3 lines of endocrinal therapy has been given in a HER2 positive metastatic breast. So Nab-paclitaxel achieves improved and targeted drug delivery to tumor by exploiting the natural properties of albumin. Nab-paclitaxel specifically binds, as I said, to GP60 and facilitates caveolin-mediated transcytosis to enhance the delivery pathway across the blood vessel to have higher concentration inside the tumor cells. And what we have understood is that Nab technology increases the paclitaxel retention in the tumor cells associated with the albumin by binding to this protein called SPARC. And SPARC is actually secreted protein acidic and rich in cysteine. Nab-paclitaxel almost has 4 to 5 times increased activity in the tumor cell versus the conventional paclitaxel. What are the advantages? There's an increased rate of transport across endothelial cell because of this Nab binding, that is albumin binding, and therefore greater and faster tissue penetration and slower elimination of paclitaxel is noted. As I said, it can be delivered in a short time, that is only about 30 minutes, without worrying about hypersensitivity, which happens in conventional formulation. And on top of it, it eliminates the need of pre-medication, which we commonly use. And especially useful in diabetics when steroid pre-medication could actually cause other issues to handle. So what have been the clinical trials in the past 6 years? We have had almost 64 clinical trials only focused on metastatic breast cancer and for Nab-paclitaxel. And this has been almost 1,600 patients. And this trials, which are only from the past 2 years, 2019 onwards, we have currently enrolled 1,600 patients on Nab-paclitaxel in various clinical trials. I'll be actually talking about this study, which is a real world data. This was done in about 168 centers in Germany, and it's called the Nabucco trial. So this was basically for 700 patients across 128 sites across Germany, and in which analysis was done in all aspects, including progression-free survival and overall survival. And this trial was basically across all age group. The average, most of the patients were in the less than 65 age group, which were around the average age being 60 years. The ECOG performance status, most of them had either ECOG performance status 0 or performance status 1, and 70% had some concomitant illness like diabetes and hypertension. This trial had metastatic breast, and the most common site, which we already know, is bone. That was seen in almost 60%. Liver was seen in 45% as a metastatic site, and others had lung, lymph nodes, pleura, or skin. Then all the subtypes of metastatic breast were used, in which HER2 positive, hormone receptor positive, and triple negative subsets were used. The most common subset was HER2 positive, which was actually, hormone receptor positive, which was almost 60% of all patients. Most of them, taxanes were used in the early stage. That is 35% had taxanes in the early stage, and almost 40% had no prior taxane use. So the people who had metastatic breast in more than one line, they probably did not had taxane use in the first line setting. 40% of all these metastatic breast cancer were in the first line setting. 25% were in the second line setting. And what we understood in this was both the treatment regimens were used, that is weekly or 3-weekly regimen. And most often, the most common outcome in all this was stable disease, as we normally have seen. And of course, the overall response rates were in the tune about 40 to 50%. Most common adverse effect noted was neutropenia, which was grade 4 in about 8%. And we had neutrophil count of less than 500 in about 4%. Peripheral sensory neuropathy was seen in again 5% in as a grade 3 and grade 4 adverse event. And motor neuropathy was just seen in 1% as grade 3 or grade 4 adverse event. So basically, the results stated that it confirmed the previous clinical trial outcomes, and it showed a favorable benefit risk profile of Nab-paclitaxel. There was a sustained global quality of life index in metastatic breast scenario, and it was effective and well tolerated even in the elderly population who had multiple comorbidities, and also in the patients who had taxane refractory disease. This was an interesting point. Clinical practice points include that it has broad range of dosage. We'll discuss about that. And it has various type of administration schedules which have been noted in various centers. Weekly regimen is comparable and probably more effective and safe than the tri-weekly regimen. It has been tried in first line setting and later line setting, and both have shown good clinical efficacy. And it was well tolerated, as I said, in less than 65 and older than 65. Most of the adverse events were manageable, and grade 3, grade 4 events were quite less. They were generally less than 10% of all types. And this now has been included in all treatment guidelines, in adjuvant settings, in neoadjuvant settings, and of course in metastatic settings. So quickly talking about the combinations of Nab-paclitaxel, we have used Nab-paclitaxel with gemcitabine combination, which Dr. Raja of course spoke about. But in metastatic breast cancer setting, we have ample evidence for this combination. Then Nab-pacli has been used with capecitabine in metastatic breast. Nab-pacli with carboplatin, again, an interesting combination in triple negative subset. Then Nab-pacli with bevacizumab. I personally don't use this combination too much, but this has been an approved combination. And Nab-pacli, of course, with trastuzumab, which we commonly use in HER2 positive subset, and Nab-pacli with lapatinib. So these are some of trials in which Nab-pacli was tried with monoclonal antibodies, mostly in the first line setting, but also beyond first line setting. And all of these data were as good as conventional paclitaxel, or in some instances superior. And these are the trials in which monoclonal antibodies were combined with chemotherapy and Nab-pacli, and again showed promising results. In all of these trials, various combinations and various regimens of Nab-pacli have been used, and the doses have ranged from 80 milligram per meter square to 100 milligram per meter square weekly to 125 milligram per meter square. Actually, some trials have tried even 140 milligram per meter square, and giving either as week 1, week 2 with a week 3 gap, or week 1, week 2, week 3 with a week 4 gap. And then again, these are the trials in which Nab-pacli was tried in the neoadjuvant settings with a good pathological CR rates, almost touching 60 to 70% in most of these studies. And then quickly summarizing the cases in which this combination, which we want to focus here, Nab-pacli and gemcitabine in metastatic breast, has been used. So caveolin 1 expression is the area of our interest in here, in which this expression was noted across metastatic breast cancer patients. And then the response with albumin-bound paclitaxel and gemcitabine was noted. And this case was, was presented in, was published in BMC Cancer Journal, in which metastatic breast in phase 2 clinical trial, weekly Nab-pacli and gemcitabine was used, and tumor versus stromal caveolin ratio was seen as a predictive biomarker. And caveolin 1 was seen as a biomarker by doing just an IHC on the stroma and the tumor cell. So basically 85 cases were enrolled in this phase 2 trial, and the results showed that the overall response rate was in the tune of 53%. And after a median follow-up of about 17.2 months, the median progression-free survival was 8 months, and the median overall survival was in the tune of 26 months. It was almost 2 years and 2 months, which was kind of good. The toxicities of all grade combined was most common neutropenia, which was seen in almost 75% of all patients. So almost 3/4 of patients had some grade of neutropenia, although grade 4 neutropenia was only seen in about 5% patients. And this was the most common non-hematological and most common toxicity. The most common non-hematological toxicity was peripheral neuropathy, which was seen in 50% of all patients, as we see in our practice. And higher tumor CAV1 ratio as compared to stromal CAV1 ratio. That means a higher tumor CAV1 versus lower stromal CAV1 was significantly associated with longer PFS when Nab-paclitaxel along with gemcitabine was given. Now, this remains an exploratory marker, and we still have to start using it in clinical practice, but definitely develops an area of interest and further research. So higher tumor CAV1 ratio and lower stromal CAV1 ratio was significantly associated with longer PFS. The second case is again the same combination, and this was presented in Annals of Oncology as again a phase 2 trial of the original innovator molecule, that is Abraxane, in combination with gemcitabine. So 55 patients were enrolled in this study, and all in this 80% had visceral involvement and 60% had more than 3 sites of metastatic disease. So NAB was given in the dose of 125 milligram per meter square, and gemcitabine was given as 1000 milligram per meter square as day 1, day 8 in a 21-day cycle. And the median duration of response that was noted in this trial was about 7 months. And of course, the progression-free survival and median overall survival was not reached in this trial. So as I said, the common toxicity which we normally see was neutropenia, fatigue, anemia. And then, of course, neuropathy, which in this case was about 8% grade 3 neuropathy. So weekly Nab-paclitaxel and gemcitabine combination has a substantial activity and manageable toxicity profile in first-line metastatic breast cancer. Further improvement can be made by addition of a biologic agent. There have been studies of adding bevacizumab as an anti-angiogenic agent. Again, as I said, there are pros and cons of using this. But these two drugs, and in fact, including bevacizumab in the combination, has shown that the progression-free survival has improved in such combination. So again, I mean, talking about the third case in which we had a metastatic triple-negative breast cancer, this study was again a comparison arm in which platinum was used. And the basic question in this trial was whether Nab-paclitaxel plus gem is superior to Nab-paclitaxel along with cisplatin or carboplatin. So in this study, AP regimen, which is Nab-paclitaxel and cisplatin, was compared in a 1:1 ratio with Nab-paclitaxel and gemcitabine. And in this, the cisplatin dose was 75 milligram per meter square on day 1 in a 28-day cycle, in which Nab was given in week 1, week 2, and week 3. And gemcitabine dose was a little low, 800 milligram per meter square, but given over week 1, week 2, and week 3. And in this, basic result showed that surprisingly, Nab-paclitaxel and gemcide was superior to the combination of Nab-paclitaxel along with cisplatin, in which the complete response rates were higher, were almost similar, but the overall responses were higher in the arm in which gemcitabine along with Nab-paclitaxel was used. So in summary, they had almost similar efficacy, and the tolerability profile was very good. And it did not favor, they said triple-negative breast cancer, platinum-based could be more effective, but would be possibly more effective with higher toxicity. Quickly, a word about triple-negative and the use of immuno-oncology or immunotherapy drug. This was IM-PASSION 130 study. This was a phase 3 study which evaluated atezolizumab, which is an anti-PD-L1 antibody, in combination with Nab-paclitaxel in the treatment of metastatic triple-negative breast cancer. Now, the dose which was used of atezolizumab in this study was 840 milligram. 840 milligram has only in the past 2 months come in our country. Until now, we had 1200 milligram. But in this, Nab-paclitaxel was used in a dose of 100 milligram per meter square on day 1, day 8, and day 15 of a 28-day cycle. And atezolizumab was given as 840 milligram on alternate week. So on day 1 and day 15, it was used. And the comparison arm was Nab-paclitaxel along with placebo. what we have to understand is that in all these patients, PD-L1 assay was done and PD-L1 CPS score was tested, in which IC, the immune cell component, was tested. And all the patients who had PD-L1 score of more than 1% had significantly better response than the patients who had PD-L1, which was 0%. So in summary, triple negative cohort has a much higher prevalence of PD-L1 as compared to non-triple negative. That is 41% versus 9% in the non-triple negative arm. And staining of PD-L1 was done in this by the Ventana kit, which is SP142, which is again available widely now in our country, which was not there till February. And this is considered as a standard companion diagnostic requirement. And there was no benefit when the PD-L1 IC score was negative. But what is also important to understand is that the higher PD-L1 score does not mean a higher response in this subset. So conclusion is that itolizumab with this study received accelerated approval by the FDA and is considered as the front line now in a triple negative with high PD-L1 score or high tumor infiltrating immune cells. So I'll end my talk with this slide, and thanks for inviting me today for this session.
Thank you very much, Dr. Aniti. Very nicely, you have taken through from the global study of like Nab-paclitaxel study, which is the entire Germany, which has encompassed 128 centers. Then the Nab-paclitaxel gemcitabine combination, then the case presentations, and the it's right from its approval study, the early stage study from Gradishare, then that first 5 years where a lot of studies got presented about the MBC, then the various types of the MBC and the TNBC. This is really interesting, and with all of you together here, Dr. T. Raja, Dr. Govind Babu, and yourself, we may look for the pan-India data in coming years, right, when all will work together. And the webinar and lockdown has taught us that without moving and without interacting physically, we can do a lot of things together. So online collections and the Panisha Biotech will be the good support in these terms to have the Indian data, as like Germany has produced, and many European countries are consistently producing their country-wise data. We are hoping that we will work together in coming future. Now, third session is of Dr. Govind Babu. I am enlightened and happy to invite Dr. Sir for the session on Nab-paclitaxel and its place in therapy of NSCLC. Dr. Govind Babu, sir.
Thank you, Dr. Kunal. Let me start. Okay.
Sir, your screen is visible, but you're not audible.
Can you see my slides now?
Yes, sir. Now it is. Sir, can you make your camera also on, sir, so you will be visible as well?
Okay. 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 Thank you. There is some problem with the camera, so it's not coming on. So I think I'll have to work without the camera.
Okay, sir. It will be nice. Because connectivity issue is there, then you continue with your PPT and with the voice.
Am I audible now?
Yes, sir. You are very well
Okay.
audible. Please
Okay.
share your screen.
Yeah, I have shared the screen. Can you see it now?
Yes, sir.
Okay.
Please
So
go
first
to
of all,
the PPT. Sir, PPT is not visible.
is it
Yeah,
now?
not yet.
Okay, it must be coming now. Now can you see?
Not yet, sir. Please keep on trying.
Okay, okay.
Earlier it was there. It was very clear.
Yeah, I know.
Yes, sir. Make a
Okay.
full screen and continue.
Yeah. Okay.
Yes, sir.
So let me again thank Panisha Biotech for getting us all together. And I must echo Dr. Raja's sentiments about Mr. Balasubramaniam. He's been a great loss to all of us. And he spoke about his singing talents. I think we should also remember his acting talents. And more than that, I recently heard one of his interviews in which he was talking about being a very good human being. I think he was an epitome for that. So I think it's very important for us to pay our respects to him and try to emulate few things, at least being a good human being, if not singing like him. So with that, let me talk about Nab-paclitaxel and advanced non-small cell lung cancer today. And this is going to be the outline of my talk. I'll touch a little bit about epidemiology, how we have evolved in the systemic therapy in non-small cell lung cancer, starting with chemotherapy and how we hit a plateau there and how we're trying to overcome it, and then Nab-paclitaxel, the studies, and a little bit about making taxanes less taxing, because taxanes are an important part of our armamentarium as medical oncologists. A little bit about Nab-paclitaxel in the neoadjuvant setting, its role in those patients who have interstitial lung disease. Does it have a role in second line in metastatic non-small cell lung cancer? Meta-analysis of all the data that we have with Nab-paclitaxel and some meaningful conclusions. So this is looking at lung cancer worldwide. And if you see the incidence and the death, the numbers are very, very similar. So which means that the mortality rate in lung cancer is very high. Again, looking at some Indian data I've shown you here. Worldwide, there are 1.4 million new cases every year, and 12.3% of all cancers is constituted by non-small cell lung cancer. What about India? We contribute about 3.2% of all the global lung cancer burden, and it ranks about 5th in terms of incidence and mortality in our country. This is some data that we have published some time back. This is a compilation of 300 patients from 2012 to 2014. This is essentially a correlation between smoking and histology. And so 85% of all lung cancer and 50% by small cell. The small cell lung cancer used to be constituted by almost small cell. We're talking about various histologies: squamous, adenocarcinomas, large cell. And the incidence of adenocarcinoma
Sir, kind request, please reshare if you are not now.
Yeah. Yes, sorry, there has been some issue with the network.
Yes,
Can
sir.
you
It's
see
okay. Yeah.
the slides now?
Yes, sir. It is very well
Okay.
visible. You
I
continue.
think there are too many webinars and there is too much load on the net.
Oh, accepted. Agreed, sir.
Okay. Okay, thank you. So this was one of the first studies of lockdown again. You know, the actually was a paper which showed that use in metastatic non-small cell lung cancer was better than just supportive care, because till that time it was just best supportive care that we used to offer to our patients, and the median survival was about 3 to 4 months. So this paper showed that cisplatin-based chemotherapy improved this, and today we are talking about survival of beyond. We are talking about several lines of chemotherapy for these patients. Various stakeholders is paying off. So this the standard backbone in non-small cell cancer is a doublet chemotherapy with platinum being an important. The initial one was empiric, then we started looking at histology and trying to base our therapies, and now we are trying to look at other markers, including a marker like ERCC1, see if that can help us choose our chemotherapy agents. Of course, soon came in all the targeted agents, and we have some targetable mutations, but the rest 50% we need to fall back on chemotherapy to bring. 75% of these are inoperable, either locally advanced or metastatic or recurrent after an early disease. And the response rates and overall survival as shown here is between 8 to 11 months. 1 year survival is 45%. 2 years. So that is where we are with metastatic non-small cell lung cancer today. So this was an important study, the ECOG 1594, because at this point of time, we got newer chemotherapy agents, which we call today as third generation drugs. So people thought that we should look at how each one would phase with against each other. And the study was, as shown here, a 4-arm study, hoping that one combination would be better than the other. Sadly enough, all the combinations, the outcomes in terms of overall survival was very, very similar. And what we were left with to decide on the combination was what was the side effect profile. So today we're talking about the evolution in treating this is how it would look. Starting with the traditional view where we had just chemotherapy, we started looking at histology, and then we started looking at newer biomarkers. And today we are talking about peak burden and several other factors. But it continues that chemotherapy remains. So to overcome the plateau that I showed you with the chemotherapy agents, people have been looking at newer modalities, and I have listed all of them here. Nab-paclitaxel. So this is Abraxane. Already heard as to this drug is different from the conventional paclitaxel. So this is a sort of a summary slide showing you all the aspects which makes this nab-paclitaxel with platinum agent with a third generation, which with Abraxane, as shown here at 100 milligrams, and that demonstrated a optimal therapeutic index with 48 free survival and a 11.3 months overall survival. I've taken this because this was their slide, which I used several. I'm sorry, I think the load on the net is really very huge. So this is the study that was published by Dr. Sozinski, and this is the scheme of the study. And this is showing you this 3-weekly dose. And coming to the clinical response, they really found meter square every week gave the best clinical as compared to various other doses. And in terms of achieving a dose intensity, which is very, that was best with the weekly dose as shown here. With albumin-bound paclitaxel, again by Dr. Mark Sozinski. This is the study design. One is, and these are the two arms, Nab-paclitaxel versus conventional paclitaxel. Secondary endpoints. Standard eligibility criteria in terms of patient characteristics. And if you look at the dose intensity, again, you can see that the dose intensity for nab-paclitaxel was definitely higher as compared to the conventional paclitaxel. Now, coming to the primary endpoint, you can imagine the investigator assessment is always review. I'm terribly sorry, I don't know what's happening to the net. So this histology. Now, in the non-squamous, as you see with squamous cell histology. So the PFS for the intention to treat population, very similar curves. The overall survival, again, very similar. For squamous, the PFS was same. But if you look at the exploratory survival analysis, this is important because a lot of our lung cancer patients are elderly. So for the elderly patients, especially with the toxicity profile of conventional paclitaxel, this could be a ray of hope. And again, showing you that in patients who are older than 70 years, the responses for nab-paclitaxel was much better as compared to conventional paclitaxel. Again, showing you the response rates and the survival, overall survival for these patients, and it was constituted of more than 70 years. So that is a significant number. So the conclusion was that inbound paclitaxel improves overall response rates as compared difference between PFS or OS between treatment arms, histology, better in squamous histology and elderly patients. And the safety was the important things that we see with taxanes is the neurotoxicity, which can be really bothersome and continue for several years after the therapy is stopped. But with nab-paclitaxel, it's been shown that this toxicity is very rapidly reversed, and therefore you can continue to use this drug. So this was approved in 2012 for use in non-small cell lung cancer. As Dr. Nidhi mentioned, use of NAP approved now, and this is a phase 2 study in lung cancer as well with the same combination. And disease control rate, which is in excess of 80% in both the arms. And the median conventional toxins and impact of solvents there. So this is sort of summarizing the whole important to use taxane because it's an important and those reductions are important. Nab-paclitaxel, this can be made less taxic than the chemophore-based paclitaxel tumor types. And this is to show you, if you, in fact, I was surprised to see this data actually national paclitaxel. I actually could not believe so when I went, in fact, true. Then the other thing is treatment for the costs that are because of the toxicity of the conventional taxanes, and this is looking at leukopenia, neutropenia, and several others, including
Looks like that Dr. Govind Babu's network
sorry
is
again,
out
I
of
don't know how
control.
many times to
Yes.
pardon
Yeah, please
myself.
continue, sir. Yeah.
So this is talking about yeah. I think so. This is about the toxicity part. So looking again at the cost of responder, we have shown that especially metastatic breast cancer. So if you figure in all those costs, you can see that the amount that is saved for nab-paclitaxel twice the difference. I think this is again important for us. This paper actually compared nab-paclitaxel, conventional paclitaxel, and docetaxel, and they've shown that the cost for conventional docetaxel is much, much higher as compared to the paclitaxel and nab-paclitaxel. So what I mentioned to you about is some special circumstances where nab-paclitaxel is in fact important, is in those patients who have interstitial lung disease, cancer patients. So this paper in those patients who have interstitial lung disease, either grade 1 or grade 2, and similar benefit can be got with paclitaxel. This is a meta-analysis of use of this drug in almost 4,600 600 patients in 13 high qualitative and safe for non-small cell lung cancer patients. Again, especially for the elderly group of patients. Medication, short infusion, and I already showed you enough data about how cost effective that is. Again, talking about thoracic radiation combined in April 2017 to 2020, follow-up is up to 2024. This is after optim 3B disease. So again, this paper again concludes whatever we've been. This is another study called Abound, which is looking at its role in second line. And because we have enough data for first line, so they will well. And this again is a randomized trial. Patients were able to withstand the Nab-paclitaxel protocols and it can be used. So there is benefit in first line, and now the study shows and overall survival as well. Again, but the tolerance even in second line was just as so. Taken together, the favorable risk Nab-paclitaxel in first line and second line is now evident. So you can use it across the first and second lines. Now, I happened to meet this person. He's
Kunal, I think we lost the connectivity fully from Dr. Govind Babu.
It keeps on getting reconnecting. We will wait till he completes his session.
yeah, I'm back again.
Yes.
Yeah, that's good, sir.
Can you see the slides? I'm almost towards the end now, actually.
Yeah, it's there, sir.
Okay. So this person is Dr. Vani, who is actually paclitaxel, and I had the fortune of meeting him at one of the Indo-US CMEs where he gave a talk. And Neil Desai, who actually worked for Nab-paclitaxel development. So this paclitaxel and for the conventional paclitaxel, giving you a snapshot of how we have progressed in treating non-small cell lung cancer. Interesting taxane in our armament long time, but is some double cost. So this is the from Panacea. We'll definitely generate more data, and it'll help many of our patients in the future. Like I was telling them earlier, somehow they had lost track for some time. Hopefully they're back now, and we'll be able to benefit a lot more of our patients. With that, I will stop and thank you very much. Sorry for all the disconnections. Over to Dr. Kunal.
Yes, thank you very much, sir. Thank you, Dr. Govind Babu, for nice elaborative session of NSCLC, right from gradation approval studies for the NSCLC till the today's upcoming studies. You have nicely elaborated, and it is the R&D scientist like Dr. Neil Desai who has invented Nab-paclitaxel, and then it has changed the complete management of the difficult to manage cancer patients of MBC, CA pancreas, or NSCLC. You are really touched upon at the end slide what is the ground reality while functioning in India. We need to present to that. So you are touched upon the very recent and highest level of the R&D scientist and the key opinion leader and the veteran doctors, along with the Nab-paclitaxel research scientist and the global first generic research scientist, Dr. Naveen Jain. And you connected all of them well. Connectivity, network connectivity is okay, but we are well connected with you, all of you. And for that, I request Dr. T. Raja to have the question answers and this moderate the panel discussion. And definitely it will be challenging to answer Dr. T. Raja. Over to you, sir.
Thank you, sir. And anyway, thank you. Nice of you to put us all together, and thank you once again for the opportunity. I'm not going to ask any difficult question. It's already a spectacular panel that we have. Doctor, very good friends of mine, Dr. Govind Babu. He's my senior role model sometimes for me.
Oh, thank you, Raja.
Definitely Govind. And Dr. Niti is a very good friend. And Dr. Naveen also, we get to learn what he has done in this field. So we will try to summarize what has been told by all of the panelists so that something like a summary and a take home, that's what I'm aiming to do. And so all the panelists, and also it could also include some of the developers in Panesha, if they can pitch in wherever needed. There are some general questions. I'll begin with Dr. Niti. She really touched upon the important aspects of difference between Nab-pacli and pacli, pharmacokinetics, how the drug was thought through in a different manner. And so in brief, what will be your important take-home message, Dr. Niti, to this question? Why should Nab-pacli be different? Why should I think it should be different, better, or whatever than paclitaxel?
So we already know about the biology, so I'll not try to venture there. But practical implications I can say is that we can deliver a higher dose at a without worrying about hypersensitivity and without worrying about tolerance. These are the two things. And in two or three clinical scenarios, which it helps a lot, is one that there's a slightly lesser incidence of neutropenia. Then you don't have to give pre-medication. So for a diabetic, it helps. And then you can give in patients who have interstitial lung disease or have had some kind of pulmonary toxicity with a conventional paclitaxel. This is what I do in practice. I mean, these things I notice, and then based on that, I decide whether I want Nab-pacli or pacli.
So one would, taking clue from you, whenever we see elderly patients, where we think about automatically somehow the Nab-pacli really flashes in all our eyes. There's no doubt about that. So then again, there has been considerable debate on how the drug evolved in their work, various trials. And there were some difficulties in finally settling down on the dose. If you listen to Dr. Hope Rugo and many of the eminent people who had worked on Nab-paclitaxel over the years, if you watch their videos a few years and then later on, you will acknowledge that they really settled down on a dose. That's how I could understand. So Dr. Niti, what will be your best recommended dose for maximal clinical efficacy and tolerance? So how do I settle down on the dose of Nab-paclitaxel?
So if the performance status is good, that is PS0 or PS1, normally I give 125 milligram per meter square. But also depends on what combination I'm using. But by and large, including the combination, whether I'm using gemcitabine as a combination or cisplatin or carboplatin as a combination, normally I try to use 125 milligram per meter square, and that's what the research or clinical data also suggests.
Dr. Govind, how happy with the dose with the Nab-paclitaxel? Where is your settled dose?
Yeah, Raja, like I said, most of our patients are elderly. So, the convenience of weekly dosing is what I'm impressed with Nab-paclitaxel. So I start with, initially, if I feel they may not tolerate, I start with a flat dose of 100 mg first.
Okay.
And if they're able to tolerate that, then the subsequent cycles, I go up to 125 milligrams per meter square. It gives me an advantage for me to monitor them each week and make dose adjustments. So I think that's what is very important for me. And in fact, wherever I use paclitaxel today, I'm replacing it with nab-paclitaxel because we have generics. It has become very affordable. And the most important thing for me is I don't need to use steroids because, most of our patients, India is the diabetic capital of the world. So if we can avoid steroids, nothing like it. So that is very important for me. And the other thing which you touched upon is its activity on the stroma. I think that is very, very important, especially in pancreas, because we know that a combination of nab-paclitaxel with gemcitabine, the responses earlier were not at all good because we did not have anything to tackle the stroma. In CA pancreas, stroma is much higher than the tumor burden itself. So, and that acts as a barrier for a therapy to reach the tumor cells. So for that, I think there's a very important drug that we have in our armamentarium. So that's
Thank
how
you,
I
Govind.
see it.
And then I think you made a kind of a sweeping statement, like all places where you would consider paclitaxel. It is almost like, pardon me for saying that, it's like a very, very sacred thread. Paclitaxel is like a sacred drug for many of the indications, and when you, with all your experience, say that you are comfortable to cross that sacred line and put it there, it's just like allowing all of us to enter the sanctum sanctorium. It's a sweeping social change you are introducing.
Yeah, I'm actually convinced with the number of years that I've used nab-paclitaxel that we can definitely replace the conventional paclitaxel with the nab-paclitaxel.
Okay, agreed. Now, Dr. Niti, coming back to you, particularly with metastatic breast cancer, the SNAP trial or the ABREST study, and or even the JAPAR 6 trial, why was nab-paclitaxel thought about when they were designing this study? Could you give us some light on what the investigators' mind were when they were actually designing this study, and what did they finally end up finding on this study?
I think so. The basic premise was the efficacy. They actually, it had higher penetration. The concept that it, as Dr. Govind Babu also said, that higher action, higher endothelial penetration, and longer, intracellular accumulation of the drug was higher. So that was the basic premise. And plus the possibility of not using cremaphore, and therefore you could give the drug safely without hypersensitivity reaction. And also when we did phase 1 studies in which phase 2 studies in which efficacy or safety with a higher dose was better in metastatic breast. That's why higher dose could be delivered safely with a higher intracellular concentration. That was the premise in which it was
So
done.
do you think they have really met their, I'm not talking about endpoints as far as the clinical trial goes, but do you think that the drug has met their expectation?
At least in terms of non-inferiority, yes. And whether it was superior or not, obviously we don't know. And I still have a little reservation, frankly, when I use in adjuvant settings. I don't use it interchangeably in adjuvant settings or neoadjuvant settings. The data for adjuvant and neoadjuvant setting we have is not as good, or the number of cases that were used is not as large. So I try not to use unless the patient's physical fitness is grossly compromised and you still want to use taxane. Only in that situation I might want to change. But in metastatic breast, yes, I use it very comfortably and
That's
very.
exactly the sacred line I was talking about. Also, in my first line use for ovary, and also there are, in our own minds, there are some areas which are considered to be very, very exclusive, shall I say. in allowing entry for certain individuals in this case now. But Govind Babu has clearly said that he could do it, he has done it, and he is more than 30, 35, and he has worked actually on the original drug. I don't know whether to call it as the original drug, let's say the innovator drug. He has done enormous work on the original Abraxane. So, words of wisdom. Now, coming to triple negative breast cancer, why would Nab-paclitaxel be special in triple negative breast cancer? Dr. Govind Babu, to you. Dr. Niti spoke about the IM-PASSION 130 study. It's really an important study, kind of a blockbuster, and changed our practice, I would say, in triple negative breast cancer, advanced disease, first line at least.
Yeah.
What are your thoughts, Dr. Govind Babu?
Raja, for me, like I mentioned, I would very comfortably replace conventional paclitaxel with nab-paclitaxel. One is because of the ease of administration of the drug. The second is the dose intensity that you can reach, which I showed you from the studies. And in fact, the initial Abraxane study that was done was comparing 175 milligrams of paclitaxel versus 260 milligrams of Abraxane. So when I actually met the initial investigators, I told them that this is a wrong trial, because you are comparing a higher dose of a drug with a lower dose and saying this is superior. So then, then finally they said we wanted to show that we could give a higher dose safely and get better responses. I really don't know how FDA approved.
Again, the weekly was kind of a finding, I would say.
Yes,
all of
agreed.
us who have been watching the evolution of this drug space
Correct.
knew that early on, they were a bulk 3 weekly and on. I think they found out of either design or by accident weekly, and it has worked well.
Correct.
Illustrating
Correct.
the point of maximizing the dose that you can.
Yeah. In fact, I remember using this when it was not available in India. One of our patients actually got it from the US, and they used to keep it so close to them because it was expensive. It was got from there, and we learned how to use it because of this patient. So I must thank my initial patient for this, for learning and getting to use Abraxane.
Conviction so far, yeah.
Yeah.
Totally agreed. And not only that, I would also say that, of course, we have known the efficacy of paclitaxel in certain tumors, but then I think this nano engineer or Nab-paclitaxel kind of really extended our confidence into some previously barren lands like pancreatic cancer.
Absolutely.
We remember, 15 years ago, if somebody has said that there's going to be very good therapeutic options in pancreatic cancers and so on, I mean, we would have looked at them with skepticism. Suddenly,
Correct.
something changed, and we discovered some biological reasons and also on and so forth. So it extended our conviction on some other untested tumors also. Dr. Niti, you spoke about spark, caveolin, and then you touched upon the tumor concentration of CAV and the stromal concentration of CAV. So do you think all these biomarker studies are really essential, or today there is clinical evidence to support our decision?
So at this juncture, these are not in clinical practice. But yes, to collect more data, they seem to be very interesting points.
What do you think about this? This year ESMO's, IM-PASSION 130 and IM-PASSION 131 together were analyzed, and I'm yet to know the complete analysis. This was presented last week on mixed results, and so in triple negative space. We know that there is a lot of interest in the triple negative with Nab-paclitaxel and atezolizumab over the last whole year. That was the hottest topic in 19 and early 20. Is there a change in the thought process, or we are okay? What have you understood so far?
The two, PDL1, we already knew CPS score is an important marker, but TIL also seems to be one thing which we'll probably be looking at. Even at the pathology level, what is a lymphocyte? What is the concentration of lymphocyte? And going forward, if there's a way of scoring TIL, that might also probably come very soon in our practice.
Okay. Dr. Govind Babu, I think you touched upon one this ECOG 1594. I'm sure
yes,
when that trial came, all of us have to memorize, know the trial, know the various arms, know the response
Correct.
rate. Otherwise, you're going to be asked about it in exam,
Yes,
and
yes.
professors and everyone was
That's right,
being on
Raja.
us. And so from that point, you emphasized that finally the toxicity and patient selection mattered, and therefore, as an extension of that thought, Nab-paclitaxel really established its role. So, what do you think is the contribution of Nab-paclitaxel in the space of CLR, particularly because you talked about toxicity as your selection criteria, but then you went on to talk about squamous versus non-squamous, and you really talked well about, even though there were some disruptions, about the meta-analysis of all the lung work that has been done. So what is your real concise message on lung NAPAC and NAPAC-LE?
So, Raja, like I mentioned, many of our lung cancer patients are elderly. So, giving the conventional paclitaxel in 1 pint of normal saline with steroids, everything many times is difficult. They also have underlying lung disease. So, all these things we need to keep in mind. So, many times, I combine nab-paclitaxel with maybe gemcitabine or carboplatin. I actually, with gemcitabine, it's very easy because again, you are giving it weekly. So carboplatin also, I sometimes divide it weekly and give 150 milligrams carboplatin weekly. So
Okay.
that seems to be very well tolerated. So I'm convinced that this is a good molecule, and the dose intensity that you can reach with weekly is really encouraging. So that's how I see it.
I would pose this question to Dr. Naveen, who has been pioneering in the development of this drug. Sir, apart from the lung that Dr. Govind Babu spoke about, the breast, various spectrum Dr. Niti spoke about, and we are now all knowing about versatility in pancreas. What other tumor group in which you are either having good data or willing to look at, work? Where are your interests? Other tumor types and all that.
Yeah, we are exploring in several other animal models like colon cancer, ovarian cancer, and head and neck cancer. And the xenograft models data are showing quite indicative response. So, so we
So
would
at
like.
the laboratory level, do you have all these what Dr. Niti pointed out, like CAV1 levels, and can you measure the spark levels? And
Yeah,
is spark very special to pancreas and that kind of a tissue, or do you see that expressed in other tissue types, caveolin, and how do you decide to choose which tumor type you want to target?
yeah, long back we have started doing some biomarker study, and we wrote one project regarding the spark expression, how to make this as a biomarker into the clinical practice. So we received also antibody to evaluate this spark level into the human tissues, and I think that project we need to reactivate. This is one of the good biomarker which directly indicate us that there is a higher accumulation, at least 33% accumulation of the paclitaxel in the overexpressed spark protein. And as you know, there are a lot of studies which published that in the head and neck cancer and pancreas, there is a higher level of this spark.
Yeah,
Okay,
Raja, in fact, the people right now in Panisha Biotech are different from whom I had interacted long ago. And we had actually spoken about this as a project across India. But then, like Dr. Jain said, it didn't happen. But it'll be a good way of actually continuing and looking at across India, across several tumor types, to actually establish maybe a proof of concept. So it'll
Yes,
be
yes.
interesting.
Good idea, Dr. Govind Babu, and I think we will reactivate this project as a
Sure.
Dr. Raja, one
Dr.
point I wanted to add here, that in lung, it seems to be a very promising drug because the data of Nab-paclitaxel along with immunotherapy agents is becoming, I mean, we have robust data. And
Yeah.
on top of it, I mean, Nab-paclitaxel is probably a drug versus paclitaxel which has less pulmonary toxicity. Lung
Yes.
cancer will normally have baseline lung functions, pulmonary function tests which are not good. So
Exactly.
this seem to be apparently safer. and plus with good robust data with, pembrolizumab, atezolizumab, which we use in first line setting. So I think the scope of this drug will increase in lung.
Right.
Yeah. So one difficult question to the clinicians, and then Dr. Naveen can answer that. So we have always had, I touched upon the pancreatic data set, and I've shown that how impact, which is a phase 3 trial, randomized phase 3 international trial, and then the LAPACT, which is actually an open label, again, a phase 2 to 3 study, which showed that, this drug is versatile in the pancreatic space. And I think the photograph that Dr. Govind showed along with Dr. Neil Desai, the third person was probably Subham Pant, if I'm not wrong. And that gentleman has done enormous work on pancreas. So now to all the clinicians and also to Dr. Naveen, that we have some, whenever we see this nanoparticle bound drug, be it amphotericin, be it paclitaxel, and be it something else, there has been always claims by several companies that they were successful, engineered, brought it. But we have unfortunately seen that a drug A, drug B, and drug C is not equal. So how do we develop confidence on this particular concept of this whole nanotechnology is convincing for us and that we can use? I'll start with Govind, Niti.
Yes, Raja, that was a great question. In fact, I wanted to ask you the same question because there are so many companies today claiming that theirs is the original Nab-paclitaxel in market.
See, I would believe it for, I would believe if they say that everybody has got pemetrexate or everybody.
Yeah, I agree.
Drugs is fine.
Exactly.
I'm worried in
yeah,
my confidence. So
that is true. It is the same doubt that I have. So then what I do is I go by the track record of the company on the other molecules that they have. That is the only way I am looking at. Because some of them with just 20 patient data have gotten approval for this drug. So I don't know how that works.
Okay. And Dr. Niti, your comment on confidence on the technology of this drug and therefore extending it to your patients.
Because Dr. Naveen presented lucidly the bioequivalent study of Paclitaxel with Abraxane. So when I'm just talking about when I'm talking about this brand, Abbott brand, I probably will be comfortable, but I don't very convenient. They are liposomal paclitaxel. I don't know what is the evidence of that. There are so many other forms of paclitaxel.
That's right.
I don't understand. So I don't use them interchangeably.
Absolutely. That's, I think rightly said. I don't pick up a drug, and once I pick up a drug, then I don't drop it kind of. So what you said is right. Something clinically, that's what I do, and I get my confidence, and then I don't allow everybody else to enter into that space just like that. So that's what.
Correct. That's right, Raja. In fact, that's why I wanted Dr. Jain to give the first talk because they have worked on it. So I wanted that insight to come in.
Yeah.
Ultimately, we are
thank
end users.
Panesia for for allowing me to do this moderation. But the last question goes to Dr. Naveen. Tell us, why should we feel confident about what is that you are presenting to us? Yes, that's all is my question. The floor is yours. Thank you.
So as we know, Nab-paclitaxel is a technology-driven nanoparticulate product. And the biggest proof of concept, I will say, biggest proof of concept is the human bioequivalence study, where total paclitaxel and the unbound and bound paclitaxel is demonstrated into the metastatic breast cancer patient. So I think we are the first company in the India which demonstrated the human bioequivalence against the Abraxane. This data has been successfully, as I mentioned earlier, accepted by the US FDA, as well as Health Canada, as well as the TGA Australia. So this gives the immense confidence to us that the particle size distribution, and zeta potential of the particles, and the in vitro release kinetics, along with the human bioequivalence, they collectively give the robust confidence that this product is exactly similar to the Abraxane.
Thank you for enlightening us. And I must admit that apart from the innovator, this is the only molecule I touch. So that's what I can say on a personal note. Um, I think I can take that liberty as a moderator.
Yeah.
But I leave it there. And excellent colleagues, excellent panelists, extremely knowledgeable Dr. Govind Babu, and very, very impressive Dr. Niti, and Dr. Naveen's great work, and Dr. Kunal and all. Thank you so much. You can now conclude us. Thank you.
Yeah.
Thank
Thank
you
you.
very much, sir, for nice moderation. Thank you, Dr. Aniti, for being here on the panel, and Dr. Govind Babu consistently supporting us in terms of this one of the big and important webinar. Dr. T. Raja, who is really backbone of the success and the nicely and so in depth, right from the Neil Desai, who has invented NAB, nanoalbumin paclitaxel, then the Gradatius studies what we have presented, and then the upcoming ongoing trial, which is the today's ongoing trial is our tomorrow's future management. And Dr. Naveen Jain, who has shown that the Indian pharmaceutical company Panacea Biotech, how has invested consistently year on year to come up with the difficult to manufacture molecule that is Pacliol. And a series of preclinical studies and the large multicentric bioequivalence study in MBC patients across the country. 17 centers and medical colleges, hospitals were involved, and we have come out with this. Therefore, definitely not the all generics are similar, and this all pharmaceutical and pharmacokinetic differentiating molecule is the choice to make the differences in the life of the patients, definitely those who are having the life-threatening conditions like NSCLC, CA pancreas, and difficult to penetrate the drug, the cancers like difficult to penetrate is the most important. I really thanks all of you, and I request Dr. T. Raja to give the concluding remarks on today's webinar.
Okay, thank you for the task, and I thank all of you. First of all, I thank the Panacea for putting us all together. I thank Dr. Kunal as the leader of the pack to think about this, drive this, and Dr. Naveen on enlightening as on what you have been through, and for the support role of Dr. Sailendra and your entire Panacea team, right from your office, head office to my local guy, Senthil Velan. He's a very good guy. I take this opportunity. I always do that too. And Dr. Niti, Dr. Govind Babu, I learned quite a bit from them today, as usual. And to all the participants and all the audience, thank you so much.
Thank you, Raja. You're always so modest. Thank you.
Thank you.
Thanks so much.
Bye,
Thanks.
everybody. Bye-bye.
Thank
Yeah.
you. Bye. We
Thank you.
conclude the session here. We can leave.
Thank you.
Thank you. Thank you.